I'm Stuart Tangye. I work at the Garvan Institute of Medical Research in Darlinghurst in Sydney, and I run the immunology and immunodeficiency research lab here. So I've been very lucky to actually collaborate with some major research groups and some pioneers in this field in the US and in Europe. And with those colleagues, we've been really active at the discovery interface of identifying new genetic causes of immunodeficiencies. So these are really cracking, finding the needle in the haystack, cracking that DNA code of what genetic defect is causing a particular immune deficiency. Clearly, when SARS-CoV-2 and COVID-19 came onto our radar over 2 years ago now, PRD patients would have been a population deemed to be vulnerable or susceptible to severe COVID-19. So I was very interested in that from the beginning, trying to assess the impact that COVID-19 was having on patients with primary immunodeficiencies. So that formed one of my research perspectives. But we also got very much involved, again, with COVID Human Genetic Effort, which is a global consortium, which has worked really hard over the last 2 years to try to view severe COVID-19, particularly in people who are otherwise healthy, as a primary immunodeficiency. And that's indeed been the case. Work led by colleagues at Rockefeller, Jean-Laurent Casanova, have really shown that a particular signaling pathway in the immune system is absolutely fundamental for our post-defense, early defense against SARS-CoV-2 infection. So defects in that pathway have shown the necessary role for that in host defense against SARS-CoV-2 infection. This has explained about 1 to 5% of severe cases of COVID-19. And then similarly, about 15% of really severe cases can be explained by people developing autoantibodies, which actually prevent this pathway from working. So that's been a huge advance. Then leverage these findings to be able to develop better treatments. And that's the case as well. We've seen the utilization of plasma exchange to lower the levels of autoantibodies that block this important innate signaling pathway. And we've seen the utilization of type 1 interferon as a treatment for severe COVID-19 in some other individuals as well. So that's really pushed along our understanding, our knowledge, both from a purely scientific, academic, biological perspective, but it's translated to a clinical outcome as well. So with COVID-19, what's been absolutely phenomenal is the deployment of vaccines, not just one, but many different types of vaccines globally. We've seen billions of doses of vaccines administered to people right across the world. The longevity of the vaccine is not as great as what we see for other vaccines. So by studying the immune response to natural infection, but also to vaccination in PRD patients, is going to be a really great opportunity to further understand how we can improve the efficacy of vaccination, what we can do to ensure a long-lasting immune response so that vulnerable people are protected against the infection, and that people can then confidently go about returning to that new normal, whatever that means, with some degree of confidence, knowing that their vaccines or their previous boosters and so on is going to protect them moving into the future.